Good morning, everyone. It's my pleasure to start this last day of the Pangenome Biohacking 2025 workshop. And it's my pleasure to introduce you Simone Rubinacci. He is nowadays a researcher at the Institute for Molecular Medicine in Finland. He obtained his PhD at the University of Oxford. Then he moved to the University of Lausanne in Switzerland to continue his research on under the supervision of Olivier Delano in the Laboratory of System Population Genetics Group. And then he moved also to the Broad Institute of MIT. His main interest in research are about genotype imputation, haplotype phasing, population genetics. And okay, I think if you are ready, Simone, you can start. The stage is yours.
Okay, thank you very much. Good morning, everyone, and thanks, Davide and the organizers for the invitation to speak. I'm going to talk today about how identity by descent methods can help to give us insights into disease mechanisms in genome-wide association studies. So in this talk, we refer to data across hundreds of thousands of people collected in nationwide cohorts called biobanks. And this is maybe slightly different to the focus of the talks we've been seeing in the last couple of days. The most famous of such examples is the UK Biobank, a cohort with 500,000 individuals, where we have genotype data, but even more importantly, large collections of well-correlated phenotypes. And the overall statistical framework to find variants that are associated to diseases is to perform genome-wide association studies, or GWAS. And the intuition behind GWAS is fairly simple. Let's assume we are interested in a disease and we want to test if a mutation is correlated with disease status. Then we can partition our cases and controls and count how many times the mutations appears in these two cohorts. And this is the reasoning behind the hypothesis testing of GWAS, leading eventually to a p-value per single variant. So GWAS have become a key tool for identifying genetic influences of human traits. And over the past 2 decades, larger GWASs have identified more and more genetic variants that associate with phenotypes. For example, GWAS of height have grown exponentially in both sample sizes and discovered associations, with the most recent GWAS analyzing over 5 million individuals and discovering more than 120,000 independent loci associated with height. So yeah, despite all these discoveries, GWAS has by no means solved genetics, but we learned a few lessons along the way. So first, we now have hundreds of thousands of variant trait associations available in the GWAS catalog that we can query. And this is a visualization of all of them across all chromosomes. We know a lot about gene programs, and there has been a multitude of publications performing GWAS. And these associations are key for risk prediction and screening tools, and they are slowly but steadily starting to be present into the clinic as well. Also, GWAS hits have a much larger likelihood to be a successful drug targets. And so several pharmaceutical companies are interested in GWAS hits for specific diseases, as they can be then focused and carried over. There is approximately a 2 or 3 times higher likelihood to be a successful drug targets than variants obtained with other methods. Another thing we have learned is that complex traits are just complex. And that's, so for many human traits, larger GWASs have pointed us to more genetic variants that associate with tinier phenotypic differences. And unfortunately, these associations are harder to individually interpret, and this is how just polygenic traits behave. And finally, we learned that there is still a lot to learn. For example, we have only a small representation of the whole spectrum of human diversity, and we miss key classes of variants. And I will come back to this in the second part of the talk. Also, as our focus is on biobank data, this focus of this talk is on cost-effective genotyping technologies. We are all familiar with high coverage whole genome sequencing that provides a view of an individual genome by sequencing the entire DNA at high depth several times, for example, at 30x. In contrast, SNP arrays is an alternative technology that has been widely used in major biobanks, especially in the last decade, and focuses on genotyping specific non-genetic variants across the genome, around 1 million common variants. And these both are cost-effective technology for GWAS analysis. However, both short reads and SNP array data do not have access to do not give access directly to the two haplotypes, the paternal and maternal component of an individual that is deployed. So we use statistical phasing techniques to actually phase the data, so to obtain the two haplotypes. And this is the process of using correlations between individuals to give us these two haplotypes. And it might seem simple, but it's a very difficult problem. First, because each heterozygous site doubles the state space, and so it quickly becomes extremely large. And on the other end, we need very large cohorts to perform this accurately. And so, of course, this makes also things even harder. So I've been involved, as David mentioned before, to a large amount of phased release of the Biobank and different data sets, SNP array, whole exome sequencing, and whole genome sequencing. And here I will focus on whole genome sequencing release that has been finalized recently. And we do have accurate method to phase SNP array data. These are the previous generation of methods, but if we will apply these methods to phase whole genome sequencing data, this will be just not feasible. So because moving to whole genome sequencing, we have approximately 1,000-fold increase in the amount of data, which means that we phase SNP array data for 500,000 individuals in one day. This will take more than 3 years to phase the sequencing data. So for these reasons, we had to develop new tools to phase whole genome sequencing data, and we call it ShapeIt5 as it extends the tool for SNP array data called ShapeIt4. And the idea behind ShapeIt5 is fairly simple. First, we phase common variants using a standard approach, essentially the ShapeIt4 approach, something that, as I mentioned, works very well and is very well established. So to do that, we build a haplotype probability model, and we initialize it with random phasing at heterozygous sites. And then we find the pair of haplotype that best describe, that best can be derived from that probability model. And this is just an initialization, and it's just enough to kick in the model. And as we keep iterating and updating this probability model, we get kind of very soon to convergence, and this will be the final phasing we will report. So this is the standard way to phase common variants using statistical approaches, and after we phase these common variants, we use this as a backbone to phase rare variants on top of it. We call these common variants as a scaffold to phase rare variants. And to do that, we take each sample in turn, and we only consider rare heterozygous sites, those that we need phasing. And for a specific heterozygous variant in one individual, we select a set of other individuals in the dataset that share long haplotype tracks, a long haplotype track that is identity by descent using the common variants that have already been phased. And we might need to force homozygosity sometimes, even if the individual is heterozygous, just because the data has not been phased yet. And essentially, we use probabilistic models that are very similar to the one I briefly described before to work up the probability that each of the two target haplotypes carry this rare allele. And we assign the rare allele to the most likely of these paths, here shown in green. And importantly, as we work with probabilities, we can also report confidence scores, and this can be very useful for downstream analysis. As we can see later, phasing rare variance is very difficult and is more prone to errors. So it's a good way maybe to filter out variance using these confidence scores. So now that I briefly mentioned how the method works, I'm gonna show you how well this performs. And this is how the method performs for the whole genome sequencing data of the UK Biobank samples. So on the y-axis, we have a measure of phasing errors called switch error rate. So we measure this switch error rate as a function of minor allele count on the x-axis, as we want to see how the method performs at rare variances, where we know that the phasing won't work as well. But actually, given the very large sample size and the high quality of the data, you can see that the method works extremely well, even for ultra-rare variants. And just to give you an example to try to understand what I try to mean with this plot, if we get the phase right in 95% of the cases with a variant with a minor allele count above 6, which corresponds to a minor allele, a copy of the minor allele every 50,000 individuals. So we are extremely accurate even at this extremely rare variant spectrum. So we are getting to a point where phasing errors and genotyping errors are start to be indistinguishable with these very large population-based efforts. So at the end, we generated a final cohort containing 200,000 individuals and around 700 million variants, all phased. So now that we have this very large cohort, it would be interesting to know what can we do with this. And there are a very large amount of things we can look at. For example, we can look in downstream application at compound heterozygotes, so loss of function on the same gene. But I'm not going to focus on that side for today. I'm going to talk how we can use this resource to actually improve other resources. So to do that, genotype imputation is probably the most common scenario, where genotype imputation is a technique that is used to infer ungenotyped variants, specifically in SNP array data, by leveraging essentially haplotype sharing into larger whole genome sequencing cohorts, such as the UK Biobank I've been showing before. So by using this as a reference panel, we can predict genotype we don't see in our data. So the most known methods to perform genotype imputation are all based on this Lee and Stevens and Markov model and share a lot of similarities to the model I've just kind of briefly described for phasing. So these SNP array imputation methods are currently extremely efficient. They cost much less than 1 cent per genome, and they can scale on the largest whole genome sequencing cohorts that are available. And indeed, imputation accuracy depends on the size of these reference panels. The larger the reference panel, the more accurate the method is. These methods are currently sublinear in the size of the reference panel, or at least sublinear in the parts that perform heavy computation. And this is, for example, what we do with the impute5 method, where we do a linear pass using a very efficient data structure called positional Borosian transform, but that after this very quick linear pass, we all have all the probability computations completely sublinear. And we recently used this impute5 method to impute the entire UK Biobank, again using Genomics England as a reference panel. Genomics England is a cohort of around 70,000 samples whole genome sequenced coming from the UK. So this is a very important resource for GWAS analysis for ultra-rare variants. And the data is being returned and is available on the UK Biobank Research Analysis platform. And interestingly, and maybe surprising to some of you, similar approaches can be used for sequencing data as well. And here I'm showing a cartoon, kind of trying to give an idea of what high coverage whole genome sequencing will look like, for example, in IGV plot, a simplified IGV plot, where a variant calling here will be fairly easy because we have high coverage individuals, especially for talking about SNP and indels in this case. However, a more cost-effective technology that has been proposed in the context of GWAS would be to decrease the coverage of this short-read sequencing data and proposing low-coverage whole genome sequencing as an alternative. For example, using 1x data, where we only have one read on average in each position of the genome. So this is what I'm trying to show in this cartoon. And this has been proposed as an alternative for SNP arrays in genome-wide association studies, but also this comes naturally in some specific application of genetics, for example, ancient DNA or circulating DNA, where the amount of DNA that is available is unfortunately very low, and we cannot do much better than 1x coverage. So with 1x coverage data, standard genotype calling is basically unfeasible, and there is a large uncertainty in the data. So how can we perform genotyping with data of this type? Well, first of all, we could model this uncertainty in a probabilistic framework using genotype likelihoods, representing the probability of the data of the reads given the genotypes. And these probabilities will be completely flat if we don't have any observed data. For example, when we have a coverage of 0, but they will be spiked towards one or the other genotype as soon as we have reads available. So the key point of this is that at a single position, we don't have much information. We have just a probabilistic representation that is maybe not particularly significant. But overall, we have enough to capture the underlying haplotypes. And therefore, using an external cohort of well-phased, high-coverage whole genome sequencing data, like the KBiobank resource I showed you before, we could use this external cohort as a reference panel and use this probabilistic assignment, these genotype likelihoods, to identify haplotypes and then perform basically standard imputation, similarly to what I've been kind of trying to give you an idea before. And by running this procedure and refining this across iterations, we could get very accurate estimates of the genotypes. And a few years ago, we proposed a method called GLIMS that solved this big computational problem with low coverage whole genome sequencing, and we made this available for the first time to very large reference panels. And essentially, the bottleneck there was that there was not there was a quadratic sampling algorithm, and for the first time, we were able to propose a linear sampling algorithm for the phasing. And we combine this with efficient data structure, such as the positional Borsellino transform. And recently, we have released a version 2 of the method where we are also introducing sparsity in the model and specific modeling for rare variants. So we have been carefully looking at the impact of low coverage whole genome sequencing to downstream association analysis and its power to association testing. And here we show that it is more powerful than SNP arrays for coverage of around 0.5x or above in every case. And even at coverage such as 0.1x, we have a very, very good power anyway. We get a power that is more than 90% that what we will obtain with high coverage whole genome sequencing. And so what I'm showing here on the left side is some local zooms that you can see the difference between 1x and different other low coverage sequencing, but also SNP array data. And what I'm showing on the right side is just an agglomerate form, just to understand what is the power overall. So we've been looking this in the context of GWAS, as I'm showing you here, but also in the context of eQTL mapping using gene expression data, and we see a similar pattern. And finally, we also look at the power of association testing for rare burden test, which looks at rare variants specifically. And even in this case, we even show better performances. Also, low coverage whole genome sequencing is better for underrepresented populations, and this is because SNP array data has to be ascertained specifically for specific populations, while sequencing is unbiased compared to the population. So this has had quite a big impact, and these results have inspired the establishment of new technologies, such as blended genome exome at the Broad Institute, concentrating sequencing efforts at exome sequencing data, having high coverage in those regions because we are interested in rare variants and rare CNVs, and offering low coverage whole genome sequencing in the remaining part of the genome, but still using the same assay, the same sequencing assay, for reducing the cost dramatically. And currently, hundreds of thousands of individuals are processed using these technologies, either in research, but also in pharmaceutical companies. They use similar assays where they genotype exome data and then use low coverage outside. So this is very likely how the future of low coverage whole genome sequencing will likely be shaped in large scale analysis. And it's very excited that method-wise, they're using the methods we have been proposing. So in the second part of the talk, I'm going to talk a little bit how similar imputation techniques and phasing techniques can be used to improve structural variant calling in large cohorts. And this goes back to actually my introduction, where I raised a last point where I said that we miss a category of variants in GWAS. And in particular, we miss structural variants, large genetic rearrangements that affect more than 50 base pairs of DNA. And they come in many forms. They are deletions, duplications, insertions. And because these structural variants are large, they can even span several kilobases. They have a large effect on phenotypes. And indeed, many structural variants have been shown to be drivers of very important Mendelian disorders. But despite this, structural variants have been rarely included in GWAS. And this means that the impact of structural variants to more like complex traits and diseases is still largely unknown. Structural variants could reveal new associations, but also be the true cause behind a cryptic SNP association, where the SNP association is an LD with the structural variant. And the reason why this has been, the reason why structural variant have not been consistently included in GWAS is because structural variant calling is technically difficult to do at scale. Roughly only 10,000 structural variants per individual can be detected from short-read sequencing data. And recently, long-read sequencing effort and a pan-genome have provided a comprehensive look of structural variation, but just for a few hundred individuals, and not in a context of GWAS. So currently, there are several data generation efforts, both in the long-read and short-read space, to include structural variants into genome by association analysis. And here we show that actually it is possible to perform improved structural variant calling with the data we currently have, so short-read whole genome sequencing and high coverage, by accurately modeling haplotype sharing in biobanks. And to be more precise, we build on previous work, and previous work has shown that from large cohorts using haplotype-informed methods, it is possible to discover and genotype specific classes of structural variants, specifically VNTRs, where we have tandem repeats, or CNVs, and uncover their effect on phenotypes. And in this work, we've been working on extending such approaches, first to detect all classes of structural variants, including insertions that are typically very difficult in short-read sequencing data, and inversions and mobile elements. And second, for scalability, extending this approach to the entire cohort of 500,000 whole genome sequencing sample in the biobank. And going a bit more into the details, the way we can detect structural variants from short-read sequencing data is to look at specific patterns in the read, such as split reads, discordant reads, or read depth patterns. And we refer these as structural variant signals in the following. And the key idea is that once we have these structural variant signals across all individuals in the cohort, we can use relatively standard phasing techniques to include structural variant signals and denoise sequencing reads, effectively allowing structural variant calling at the haplotype level. So we developed a method for this, it's called SUVI, and to perform such analysis in the UK Biobank. And in short, the method has three main components. First, we detect structural variants, then we extract these structural variant signals across all the CRAM files, and then finally we perform haplotype code. We perform coding of structural variants using haplotype phasing. I'm not going to go into the details, so now we perform detection to the sake of time, but the key idea is that we can use the structural variants that are known, but also we can perform structural variant discovery by just binning the genome and looking at the structural variant signals. And we have a way by looking at siblings, a correlation of IBD2 segments, so segments where siblings share, they are monozygotic twins in this region. We could look if there is inheritance, if these patterns, if these signals are shared, then it will show that this is inherited. So once we detect the structural variants, then we need to extract the structural variant signals across the entire cohort. And this means processing 500,000 high coverage whole genome sequencing CRAM files. It means processing around 10 petabytes of data. So this, given the size of the data, this is of course the most computational intensive part of the analysis, but we heavily optimize this step and we are able to process the entire cohort for less than £1,000 in the UK Biobank search analysis platform. And finally, once we get these signals, we can phase them and denoise them using information across haplotypes. And at the end, we have this variant level cohort that can be directly taken to association testing. And the UK Biobank has a very large large phenotypic cohort, so we can really test several phenotypes. So in our analysis, we also included European individuals from 1,000 genomes, a project to compare our structural variant calls with the available short reads and long read sequencing data. So we use structural variants derived from long reads that are available for a subset of individuals, and we checked how well short read methods, our SUVI approach, but also what is available from 1,000 genome using short reads, I've been able to detect these high-quality structural variants. And we found that our approach detected more high-quality structural variants than it was there with 1,000 genomes. So after assessing this, we wanted to do association testing, and we did association testing using 57 quantitative traits in the UK Biobank. And our goal is to identify structural variant associations that are not explained by linkage disequilibrium by any nearby SNP indels. And we recently completed this association analysis. We are still finalizing some file mapping. And in the following, I'm showing a couple of examples just to give you an idea of the structural variance association we find, how they look like, and what we can do with them. So first, we identified a 2.3 kb deletion on chromosome 3 linked to liver biomarkers. So this deletion is also present in ROMADSV, and it's similar allele frequency in Europeans to what we detect in the UK Biobank. This deletion is strongly associated with 4 liver biomarkers, having the highest association with alanine aminotransferase, and having an effect that is 1.4 times stronger than any SNP or indels nearby. So we tested the variant association for liver disease, and we indeed verified that there is a borderline association for liver disease as well. So we believe that this variant is a strong candidate for follow-up and replication analysis in our cohort, and we are currently carrying this in all of us. Another interesting locus we identified is a VNTR in the second intron before GLT1 on chromosome 9, and it is strongly associated with serum levels, a biomarker of diseases like gout. And its association is nearly twice as strong as any nearby SNP indels. So this VNTR has a 39 base pair repeat unit and is extremely variable across individuals, with common alleles having 1 to 6 copies. And these 6 alleles are also visually present in high-resolution assemblies from the Human Pangenome Reference Consortium. So we observe an allelic series for this VNTR, where longer VNTR alleles are linked to higher rate levels and greater gout risk. And the best tag SNP for this is a topic UTL for before GLT1, suggesting that the VNTR might affect gene expression. So these findings kind of motivates us to study, kind of use GTEx to find map this eQTL signal and explore whether this VNTR can influence splicing. So to summarize, the methods I've shown you today provide an overview on how shared patterns between individuals in large-scale biobanks can help informing us of disease status, disease genetics. However, we are far from done. We still have, we only explain a small fraction of these trait associations, and there are many directions that remain for further work. As many speakers have been mentioning in the last couple of days, we won't be able to completely assess the effect of structural variants using long-read structural variants until we have long-read sequencing data. And some of the algorithms that have been presented in the last couple of days will definitely help to reach this goal. And we need more data and better data. This also means that it's a great time to be a statistical genetics, and we will have a lot to discover in the coming years. And in Finland, we have a fantastic data set, which is FinGen, with many potential new discoveries in the years to come. And this concludes my talk. I just would like to mention that I'm currently looking for PhDs or postdocs to join my lab. So if you're interested in this type lines of research, please feel free to contact me. But also, our institute has a fantastic PhD program that allows rotation between several labs, and it's a great opportunity for students, and the deadline is just in 2 weeks. So go and check it out if interested. Thank you very much for listening. I'm very happy to take any questions.
Thank you so much, Simone. For the audience, I remember you that you can post your questions on the Matrix chat. And for Simone, we have about 20-30 seconds of delay between our meeting room and the live stream. So maybe you can wait. In the meantime, I've just 2 questions. Both of them are probably very silly, but the first one, regarding the low coverage approach, probably I missed the point. Do you have a sort of threshold, a lower bounds under you're not able to perform very well, or?
Yeah, that's definitely true. That threshold will be probably around 0.1x. I think under that, when we tested, things will start to become very difficult. So it's still usable to some extent, low coverage with genome sequencing data less than 0.1x, but it will require probably even larger reference panels, or maybe imputation is not the best approach because we start not being able to capture haplotypes anymore, at least with the current methods we have now. But until 1x, especially if we have a good reference panel, we can do extremely well, and that was maybe surprising, and I wasn't even expecting it myself when I started working on this.
It's very surprising. I agree with you. The second question is just out of curiosity and more in the context of this workshop. Are you aware of any approach based on pangenome graph, or I don't know, GWAS or some topics related?
That's a good question. I'm sure that there are people thinking about this. So now maybe more in the context, more than GWAS, more like how to use the pangenome graph to perform imputation. And I think that's definitely there are some methods that perform this in the context of maybe not pangenome directly, but personalized reference, essentially. So the idea is to perform imputation and then create your own personalized reference, but now you have the alternative alleles in your personalized reference, and then remap your reads essentially into this personalized reference. So definitely there is I'm confident that there are many people thinking about this very carefully, and potentially also in the context of pangenomes. But I'm not sure about connecting this directly to GWAS, because, well, of course, when the pangenome will give us access to darker regions of the genome. And so that's definitely something people will want to do to understand what is the impact of this structural variants and this dark region of the genomes in terms of phenotypic effects.
Okay. Thank you again. I don't see any question on Matt's chat. Let me check another time. I don't have any question. If I miss some question, you can just post them later and we will forward to Simone. So thank you so much again. Now we will have